Good afternoon, everyone. I'm happy to be here with you virtually for now for an update on local and national research efforts. As a little bit of an introduction, I am a research assistant professor at the University of Pittsburgh, but I've also been very involved with the ALS community. At the local level, I am the past chair of the beloved Western Pennsylvania chapter, and at the national level, I serve on the Research, Care Services, Advocacy, and Communications committees as a national trustee. And in all of these efforts and all of these positions, my goal, much like you, is to realize a world without ALS. So again, I'm happy to be here with you for now virtually, but eventually for the live question and answer session. So I'm going to start with some key features of amyotrophic lateral sclerosis, or ALS. And these features that I'll bring up are to highlight some of the research developments and the research efforts that I'll go into both at the local and national levels. So we know that ALS is a heterogeneous disorder, and it involves the loss or the death of motor neurons. And motor neurons are a specialized type of cell that are directly responsible for controlling muscle movement. And these cells are located in particular areas of the body, in the brain, in the brainstem, and in the spinal cord. And I'll touch on that in more specifics in a few slides. We know that both sporadic or unexplained disease causes of ALS exist, and that there are also genetic causes of ALS. Although the vast majority are sporadic, we can learn a lot from the genetic causes. So for example, 20% of genetic causes are as a result of mutations in SOD1, and this is a model that's used in many systems, and in fact I've used it and currently use it in my research efforts. An additional example is the NEK1 gene. Mutations of that are responsible for about 3% of genetic causes of ALS. And this is not an exhaustive list, but we'll touch on the genetics when we talk about the national research efforts. Unfortunately, at this point, besides those genetic linkages and some of the general processes that are associated with it, we don't know what causes ALS. And so what happens in ALS in this cartoon, just to orient you, the blue here is a motor neuron, and a motor neuron receives inputs, and so that's shown by these red arrows that connect signals from all of these other cells, and it passes along its information through its axon, which connects, in this case, to its target muscle fiber. And so this connection occurs at what's called the neuromuscular junction. So the neuro is the neuron, and the muscular is obviously the muscle fiber. And this junction is really critical because it's this junction that's lost in ALS. It no longer works. So the motor neuron is lost, and it's unable to communicate with its target muscle. So the muscle can't be controlled. And so it wastes away and is unable to be used to perform the tasks that we'd like it to perform, that we're used to it performing. And in this case, or in this picture, you can see another cartoon that's giving us an example or a variety of examples of the causes or the potential causes underlying ALS. So in this case, the motor neuron is shown here in green. And again, all around it are different things that are thought to contribute to the disease process. So I've mentioned different genetic factors. There are environmental exposures that may play a role. There are some other cell types within the nervous system that can contribute to inflammatory processes. And all of these things together can lead to what's known as apoptosis or cell death. So that's the loss of this motor neuron. And the one thing that I want to bring up here that's thought to play a role is impaired axonal transport. So you'll notice all of these green processes that are going out from the neuronal cell body. And you can think of these as highways that allow the cell to not only receive information, but to pass along information. And that information can be in the form of proteins that travel from the cell body all the way down these highways and then are released to the target cell. Or it's just the maintenance of things that travel back and forth along these highways. So this is an important process within the cell, and if it doesn't work correctly, it can have dramatic effects on the overall function and health of the cell. And so now what I'd like to do is just touch on some local research efforts. The first relates to some work that I am currently doing in my laboratory at the University of Pittsburgh. And then the other two are some efforts that I have been involved with, some ongoing and some that are sort of moving forward in different directions based on some work that I've done in the past. So the first, understanding ALS initiation and progression, is work that I've been doing at the University of Pittsburgh. And I first want to start by acknowledging my team at the Systems Neuroscience Center, and of course the funding that we have received from these various agencies. But one thing I want to point out to you is that this is the team that I am privileged to work with, but it's also a part of a greater team at the university that's focused on ALS and ALS-related conditions. And they're part of the Center for ALS Research. So here I've just listed some of those investigators as well as their team in general. This is certainly not an exhaustive list, but I just want to let you know that there is a lot going on at the University of Pittsburgh, and we do our best to collaborate across these groups and to meet relatively regularly with each other so that we can stay up to date with what each other is doing, and also to help each other's labs realize their goals and to find ways that we can collaborate. So as I mentioned, ALS is a motor neuron disease, and that means that the motor neurons, those specialized cells that control muscle function, are affected. And those cells are located in very particular areas of the body, in the brain, in the brainstem, and the spinal cord, and that's denoted by these orange arrows. But one thing I hope that you can start to appreciate is that each of these blue dots is representing the location of those cells, but also that there are a lot of connections that can be made between them. So a cell in the brain may connect to another neuron in the brain stem, which may connect at one location in the spinal cord and then to a lower location in the spinal cord, thereby forming this motor network, or this network of connections that is important. So at each step along this pathway, there's a communication node. There is an exchange of information that is critical for the overall function of the system. And so when we get down to the spinal cord, that is where the connection between the motor neuron and its target muscle occurs. But everything else that is included in that network, from this connection all the way back up to the brain, is part of that network. And so it's important for us to understand how this network functions in normal conditions, and then to understand what might go wrong in the case of ALS. And so back to those questions, those big questions of disease initiation and disease progression. If we work our way back, if we start at the muscle and work our way back through that complicated network, what we can do is, again, understand what it looks like in normal cases and try to see if we can determine when something first goes wrong in the case of ALS, and then give some clues or deduce some clues to help us understand the progression of the disease, again, throughout that motor network. And so we do that using a tool, rabies virus, which is actually a tracer. So we can actually, and when we say tracer, we mean if we inject it one place, we can sort of follow it along the connected pathway of neurons and understand which neurons are part of that total motor network. So in this case, again, we're starting at the muscle, we're starting at that point, and we inject rabies virus into the muscle. any cells that are directly connected to that muscle, so this would be motor neurons that are directly connected to this muscle, will eventually take up the rabies virus, and again, via those highways, transfer it back to that motor neuron. From there, the virus will replicate within this cell, and then any of the cells that are directly connected to this cell will then take up the rabies virus at these different nodes of communication, and via their own highways within those cells, transfer that rabies virus back to those cell bodies. And then this keeps occurring in a time-dependent fashion. So we can vary the timing and we can get more or less of the network that we get rabies virus to trace based on that timing. And importantly, what you'll notice is any neuron, any motor neuron that's not directly connected to that muscle will not take up the virus. So we're really just looking at the motor networks that are associated with a muscle of interest. And so what this allows us to do is, again, start at the muscle, so down here, and then we can inject the rabies virus into the muscle. It'll travel back to motor neurons in the spinal cord, to inner neurons in the spinal cord or the brain stem, and then to neurons within the brain. And I have a couple locations here, but these are just layers within the brain that these cells are located. And this allows us, again, in a simplified version, and this is sort of something that we use as a summary of our work. We pick muscles of interest. So in this case, muscles that we know are affected in the animal model that we use of ALS. We inject rabies virus and via time-dependent manipulation, look at motor neurons directly connected, that are indirectly connected via inner neurons, and then up into the brain, which are longer, they're further away distance-wise and connection-wise from this original target, the muscle. And so as an example of what we can see is after we inject rabies virus into our target muscle, we can look at the brain and see if we see cells that are labeled with rabies virus. And that's exactly what this image is showing. So here, all of the blue is just a marker for the nuclei of cells. So there are tons of cells within the brain and in different layers of the brain. But what I've highlighted in this box here and what's blown up here, the green cells are cells that have been traced or labeled with rabies virus. So these cells have been labeled after we've injected rabies virus into a muscle. We've allowed the animal to survive long enough so that the rabies virus can be transported all the way to the brain through that motor network, and then we can determine where exactly those cells are located that are connected to that muscle indirectly in the brain. And so what we do after these injections and after looking at images like this is create maps. So I know this doesn't really look like a brain, but it's how we represent the half of the brain that we are looking for. So once we inject into a muscle, if we inject to a muscle on the right side of the body, we look at the left side of the brain and look at where those cells that are labeled with rabies virus are located. And so this is an example of the maps that we create, and the red and the shades of red are just indicating the density, so the number of cells that are labeled with rabies virus in each of these locations. And what we can do is understand the location of the neurons that are connected to each muscle that are part of this motor network, and then we can compare them. So we can compare the maps from different muscles. So here's one muscle, and here's another muscle, and this is the same time point, but you can see there are dramatic differences in the number of cells that are labeled. And so we can compare not only the different muscles, but we can compare different disease stages. And we can start to understand what changes and understand where those changes occur within the motor network and when they occur as the disease progresses. So this is ongoing work. And so with our studies of ALS, as I mentioned, we can compare the maps to determine what's different and how different muscles might be affected. And importantly, we can understand the timing. So when do we see a change, and is there a delay in the transport process? And is that really what's underlying the death of motor neurons that we see in ALS? And a couple other things we can point out is that there are different neurons in different parts of the motor system. So can we start to tease apart how these might be affected differently? And then we might need different therapeutic strategies or different drugs that would help each of these types of neurons. And then for the transport, again, along those different highways, we want to understand if these highways are basically a traffic jam, so nothing can get through, or are these highways a conduit that provides an avenue that lets the disease propagate throughout the motor system? So we're working on all of these questions now, and we hope to have answers in various different model systems so that we can compare across the model systems and really get a better understanding of this disease, and then a way to treat each of these populations of neurons and the motor network itself effectively. Okay, so the second area that I want to touch on is some work that I've done in the assistive technology space. And this has been in collaboration with groups at the Rehab Neuroengineering Labs and within the Swanson School of Engineering, again at the university. So Steve Gleason has been quoted as saying, "In the absence of a cure, technology is the answer." For a variety of reasons. Access to technology, as we all know with our smartphones and smart devices, can improve quality of life. And for those that have muscle loss, can provide greater independence with abilities to not only remain engaged, but to do things that are now difficult or they are no longer able to do because of the loss of that muscle function. And so you can think of this one aspect or one area of technologies is the area of brain-computer interface technologies. And in these technologies, you can think of the brain and the cells within the brain communicating and controlling external devices. And so really, I think of this as listening to neurons. So when we listen to neurons, we use these, they're called electrode arrays. So this is what it looks like here, and it's very small. So here it is pictured here compared to a penny. So just a small fraction of the size of a penny. And what happens is these arrays or these electrodes are implanted into the brain, where they're able to hear the electrical signals that are, or record the electrical signals that are released by the neuron, and then to translate these signals to an effect. So down here in this cartoon, what the image is showing is a person that's mentally visualizing both hands closing, although one hand is closing and is connected to a robotic arm. And so with that visualization, the neurons are firing and sending electrical signals. These devices can pick up those signals and then translate those to an effect. And although this might sound very science fiction-y, it is actually happening. And this is again in collaboration with those same groups. This is Jan, who has a disease that resulted in her basically being paralyzed from the neck down. And she has two of these devices that are implanted, and they are connected here via this gray connector to this robotic arm. And using her thoughts and her visualization and thinking about moving her own arm, she is able to control this robotic arm to feed herself a chocolate bar, which is what her goal was when she started this study. So these are great technologies, but the problem is that these are hard metal surfaces, and these hard metal surfaces just don't fit into the environment, say for example, of our brains, which is a nice, soft, pliable environment. So some of my efforts have been focused at making the response, so the brain's response to these hard metal devices, making it a more favorable interaction so that they could be clinically viable for a long period of time. And we've done this in a variety of ways, but we've tried to basically modify these surfaces with different approaches, and then also been involved in designing just softer materials that match more of the environment of the brain and the biology of the brain. And so I'll just show you a couple examples of what modifying the hard metal looks like. Again, this is that hard metal electrode, and this is one of the modifications that we've done. And you can see in this picture, there's a lot of contour and a lot of gradient to this modification. And actually neurons really like this type of environment. So I'm just going to highlight this panel right here with the red. And so this is a surface, just the plain hard metal surface, and this is the surface that's modified to have this additional contour and texture. And you can see that these red, large red globs here are neurons, and they're very close to this electrode that we've implanted. So that's good. That means that the electrode can actually listen to these neurons and record their signals. Similarly, when we used a softer material, we had the same sort of trend. So in this case, on the right side here, and I'm denoting the area that would be associated with the electrode in white here, this white circle. So around where this electrode would have been, when we have the stiff metal, we see an area that's largely lacking of motor neurons. But in this case, with the soft material, you can see that neurons are very close to the site of this electrode. So this is a very positive result, and this will help to improve the overall performance of these devices, such that once they're implanted, they can last the lifetime of the person for which they've been implanted. But as a result of my involvement in these assistive technology endeavors, I was able to help initiate and participate in a roundtable discussion about 5 years ago on incorporating more assistive technology aspects for people living with ALS. And so this was in collaboration with Prize for Life with the ALS Association. But the goal was to help people with ALS continue to have their voice. So it wasn't just the technologies that I've just outlined, but any types of assistive technologies. And this led to the launching of the ALS Assistive Technology Challenge, and the idea that the ability to speak is so important. And so giving back the ability to speak, the freedom to have that speaking ability is so important. And so here are some examples of assistive technology studies that are ongoing today. Not only clinically useful brain-machine or interface control of robotic arms like I outlined, but 3D printed masks for use with non-invasive ventilation, banking voice to use for software that can be utilized by people with ALS, utilizing power positioning of speech devices and other equipment, and then efforts in telemedicine with brain-computer interface technologies. And so I just want to highlight that this continues to be a funding priority of the ALS Association, and something that we feel is a very important thing to make sure we continue to develop. And my final personal story goes from the identification of biomarkers and proteomics to promising therapies. And this I did first as a graduate student, and then in collaboration with Robert Bowser and his team at the Barrow Neurological Institute. So first, what are biomarkers? Some of you may have heard this term thrown around before, but this is really anything that can be used as a marker of a disease process. So you could identify biomarkers for people who are susceptible to getting a disease. You could develop biomarkers that are useful in diagnosing ALS or other diseases, things that might indicate that a disease is progressing, or in the case of drug development, something that indicates that a drug is actually working, that it's having the effects that we want it to have when it's administered. And so in my work with Dr. Bowser, we were attempting to identify biomarkers that could be used to diagnose ALS. And one of those was a protein called transthyretin, or TTR, is what you'll see here. And in ALS patients, it was decreased relative to not only healthy controls, but a variety of other neurological conditions like Alzheimer's disease, multiple sclerosis, and then other dementias. And this protein, so it's shown here, impacts the signaling of another set of proteins. You can see that this starts to get complicated very quickly, but what I'll just say is that this protein affects the expression of genes within the cell. And so these genes can impact overall function and the overall health of the cell. And what we found is that this signaling pathway that was impacted by our biomarker TTR, or transthyretin, seemed to increase the amount of a particular signaling pathway in sporadic ALS. So this red here is within the nucleus of a neuron. And you can see it's much more dramatically, it's dark and dramatically stained here compared to a control neuron or a neuron from someone with a genetically linked cause of ALS. And when we looked at this in cells in a dish and we stimulated this pathway, what we found was that it was protective. So it was actually a good response that these neurons were having. It was protecting them from undergoing cell death. And so the exciting thing about this was that if we could develop drugs or a system that we could stimulate this pathway first in animals and then in people with ALS, perhaps we could protect their motor neurons from being lost or dying and keep them healthy and then, stop the disease process. And so those are the studies that have been done now, at least in animal models. So what we've done is administered a drug to mice, and this drug stimulates this signaling pathway. And in this case, when we administer the drug, we see blue that's shown and expressed in the nervous system, and that just means that it's working. So we're able to actually stimulate this pathway. This is with the drug here and without, and you don't see any blue without the drug, so that's good. It's a specific response. And then we did some tests in the mice. And so with the drug, we were able to increase the survival of animals with ALS. And with the drug, we're able to improve their function. So their performance on locomotor tests and their performance on strength tests. So all positive responses in animals with ALS. And if you'll remember back to this cartoon, that it's important that this connection between the motor neuron and the muscle stay intact, and that we have these motor neurons that are not being lost. And so with the drug, here in green, every one of these green marks is a motor neuron. So we have more motor neurons when we have the drug compared to without the drug. And we're keeping these connections maintained. So the neuromuscular junctions are not being lost. They're preserved with the drug. So on the bottom here, this is with the drug. So you're seeing this blue, which is the motor neuron, and then the red, which is its connection to the muscle. So those are maintained here, and without the drug, you don't see that. You see very little and a loss of those neuromuscular junctions. Finally, with the drug, there is less inflammation. So here in green here, this is without the drug. You can see a lot of green, and you can see much less green here with the drug. The same can be said here. In this case, all of these red dots, each of these red dots is an activated microglial cell, which is associated with inflammation. So without the drug, you see many more red cells than you do here without the drug. So overall, a reduction in inflammation. So this drug had a lot of positive outcomes in the animal model, increasing survival, increasing the number of motor neurons, maintaining those neuromuscular junctions, and decreasing inflammation. So it's now moving and being modified for delivery in humans to the central nervous system. And so, somewhat dependent on the form and the chemistry that ends up taking place, there are already similar drugs that have been used in humans. So we're hoping that approval for these trials and then the subsequent studies can move more quickly. So this is an exciting area, and I will continue to keep everyone updated on this if you're interested. So now I'd just like to spend the next 15 or 20 minutes or so on national research efforts, giving you a sense of what that portfolio looks like, some of the collaborations, and then just ending with a few success stories, things that we're really excited about in the field in general. So the important or the exciting development can be seen here, which is in 2013, before the Ice Bucket Challenge, the National ALS Association contributed about $6 million per year to research efforts. And last year, it was about $21.5 million. So such a dramatic increase as a result of the funds received via the Ice Bucket Challenge. And so post Ice Bucket Challenge, some of the things that have been on the forefront are global collaborations and making things open access, increasing the investment, and then really identifying more genetic causes. There's been a real increase in that since the Ice Bucket Challenge. Forming multidisciplinary teams and really getting a lot of partners, not just industry partners, but people within the technology space and in government to really commit to some of the challenges that we're facing and addressing those with us. And so I would like to also highlight that the ALS Association is the largest philanthropic ALS funder in the world. And so with the research arm of this three-pronged mission, the Ice Bucket Challenge allowed us to really commit significant resources in this area, over $110 million since 2014. There are over 170 active research projects in 15 countries. And a lot of the funding has been for infrastructure, for training, for education grants. We don't fund things like a new building. It's really just in the education and the work that needs to be done to tackle this disease. And another key point is that the scope and magnitude and mission. So the mission, not only research, but care services aspects and advocacy, all being integrated. And then the leveraging of these funds to bring in additional partners. In terms of operating principles, which I think are important, is to really steward and set the agenda for the ALS research space, to focus on novel and innovative work, and to really give it a boost at critical junctures. So times when there's not any other mechanism to get that technology or that drug or that scientific project up and running, the ALS Association really works to try to give it a boost at those critical junctures. Leveraging the investments and broadening our impact, building partnerships, and then also participating in things that are non-funding support. So a perfect example is that assistive technology roundtable that the ALS Association sponsored, that ultimately led to assistive technology being a funding priority of the association and the field in general. And of course, we want to operate with integrity, transparency, and accountability. So there's an extensive peer review process for anything that's submitted to be funded. We want to make sure that the areas of care, services, and advocacy are integrated, and that anything that we fund in any of our initiatives can always be accessed on our website. So if you haven't checked that out, please do so, and you can see by research area, by person, and the amounts that they've been funded and some of their progress. So it's a really nice way for you to stay up to date on things that have been and are currently funded. And then accountability. So we really want to make sure that anything that is discovered is published, and that any data or resources that are created are shared with the entire research community. And so some of our strategies, not only maximizing and leveraging our investments in research, but attracting young scientists, so fellowships to promote their entry into this field, and then inspiring partnerships, so really driving collaborative efforts across the research field. So some of those efforts include some of these strategic initiatives. So there are ones with the New York Genome Center, for example, Project Mine, Answer ALS. So these are consortia that include multiple universities and multiple investigators that are all aimed at not only genome-wide association studies and identification of genetics, but also therapeutics and things related to ALS treatment and related disorders. And I'm just going to go through some of the partnerships. So these are some partnerships with academia and industry. So some of the companies that we're involved with now, including big ones like GlaxoSmithKline, Iron Horse Diagnostics, Ionis Pharmaceuticals, which I'll touch on a little bit later in some of their work. Partnering with other nonprofits within the ALS space, but also outside of that ALS space. So the Packard Center is located at Johns Hopkins University, and then there are the Muscular Dystrophy Association, which you're probably familiar with, and ALS TDI, and Team Gleason. So we try to partner with all of these associations, and then as well with the government. So you'll be able to hear Dr. Mehta talk about the CDC registry and all of the collaborative efforts that have been successful with that effort, and really focusing in on some of the advocacy efforts here. But with all of these organizations working to benefit the entire ALS community. So what I just want to stress is that this is really a multifaceted approach. So not only are we trying to address basic science and identify risks and causes of ALS, but we're trying to move towards drug development through the different phases of clinical trials, and understanding and managing ALS better. So you can see assistive technology is here, but there's also the importance of impacts on families and caregivers, and the access to quality services and natural history of the disease. So prevention approaches. So a lot of things that are going into the research program that are important. And so what I'd like to now touch on are just some successes, and it's certainly not led completely by the ALS Association, but in their partnerships throughout, and then the developments from a national perspective. I think that we've been able to not only leverage but promote these developments over the course of the last few years, especially. So the first is just with respect to the genetics of ALS. So you can see with this graph of gene discoveries linked to ALS, and these are those supported directly by the ALS Association. In 1993, we had the first known genetic cause, which was mutations in SOD1. And then it took till 2006 and really till 2008 for the TDP43 mutations to be linked to ALS. And you can see that there were a couple more, but then you see in 2014, again, when the Ice Bucket Challenge hit, and then the real increase from that, and this is a little bit dated, only going to 2018, but the significant increase in the number of genes that have been identified. This just looking at 4 that were discovered since 2016. Again, I think this one is dated a little bit as well, but you can see that there has been a significant increase in these genetic linkages to ALS. And so the importance of genetics is that it helps us to understand maybe what more of those initiating factors are, and also some of the pathways that are involved in the disease. So as a result of the identification of genetic causes, we know that RNA processing is an important common feature of some of these genetic mutations. Protein degradation. So when there's too much protein that's hanging around, it sort of clogs up and prevents the cell from functioning normally. And it can't do that. And so then it gets sick and then it dies. Cytoskeletal dynamics are another one. And these go back to that idea of highways within the cell that are trafficking things and information up and down the cell body and down the axon. And so the fact that this is important suggests that mutations in certain genes that impair this process are going to be detrimental. And I would like to say one other thing about the genetics of ALS is that it helps us create model systems. So whether the model system is a mouse or a rat or a fly or a worm, regardless of what model system we use, these genetics allow us to create those model systems and to study the disease. And so when it comes to genetic forms of ALS, there's one approach that is very exciting. It's the antisense approach, or ASO approach, antisense oligonucleotide, ASO. And again, the most promising targets are forms of ALS that are linked to genetic mutations. So that includes SOD1, C9, ORF72 cases, and TDP43 cases. And so I'll just give you an example of what this might look like. So in a normal neuron, the brown here is the protein of interest. In this case, it's TDP43, but the brown here is where that protein should be. But in ALS, that protein is not localized where it should be. It's somewhere else, doing something else, and it's causing harm to the cell. So what happens in this antisense technology, again, in normal conditions, and this is going back to some maybe high school biology or maybe some college biology for some of you folks if you took it then, but we have our DNA, which includes all of our genetic material, which is transcribed to mRNA and then translated to protein. So if we were talking about SOD1 protein, in this case, that protein would be made normally, even if it was the mutated form that's doing negative things within the cell. So the theory behind the antisense drug is that if this is introduced, then it's introduced at this stage. So once the mRNA has been formed, the antisense drug comes in and binds to this mRNA and destroys it. And so none of the SOD1 protein is made. So none of the mutated SOD1 protein is made. It can't clog up the cell and have negative effects. And this therapy is really promising. It's already shown promise in something related to ALS, spinal muscular atrophy, which is something that affects children. It's been called ALS in kids, and it's had some really significant and dramatic positive effects there. And so in ALS, this is the VALOR study, and it's currently in phase 3. You can see that here. This is the tofersen VALOR study here. And this gives you a sense of some of the other clinical trials that are going on. This was updated in May 2020. But those that are in phase 1, which is the earliest phases, phase 2, and then phase 3 studies. And all of those that are listed with the 2 asterisks indicate those that have been supported by the ALS Association. So this is really encouraging because there are 32 interventional and 34 observational studies that are ongoing. And what I'd like to highlight now is what's listed here in the phase 2, the HEALEY platform. So this has been pioneered at Massachusetts General Hospital. And the beauty of this platform is that it tests multiple different drugs at the same time and uses one common placebo group. So in essence, this leads to less people having to be on the placebo and more people actually receiving a drug. So you enroll in the general platform trial, and then there are multiple different drugs that are being tested at one time. And all of these drugs are sharing infrastructure. There's the same protocol that's underneath all of them. And so because everything is the same and this shared structure, you can share the placebo participants. So what that looks like is for drug A, for example, out of the 4 groups that they've shown here, 3 are receiving drug and 1 is receiving placebo. The same thing for drug B, 3 of the 4 groups are receiving drug, 1 is receiving the placebo. Drug C, 3 groups receiving the drug, 1 placebo. And so what we can do is pull all 3 of these placebo groups and share these to compare to all of these active groups. And so this is far superior to the traditional clinical trials. It not only cut costs and cuts time, but again, it increases the number of people actually receiving a treatment versus those that are receiving a placebo. And so I'll just sort of go into some of the current efforts in the field, a summary of those. So national research efforts are aimed at a diverse number of biological targets, so various genetic causes, but also some immune components like the Tregs, which is a type of T cells, and decreasing that immune associated reaction. There are multiple drug approaches. So I talked about the Antisense approach, but there are also other antibodies, stem cells, and small molecules being investigated. Engaging in partnerships with not only the industry, but other nonprofits, investment groups, and the government. Increasing clinical trial efficiency. So I mentioned the HEALEY platform, but also working with the FDA and the Northeast ALS Consortium, or NEALS. And then, of course, robust efforts in just understanding ALS. So not only genetics, but basic science efforts and community engagement. And one thing I just want to point out here, ALS Focus, which is relatively new. It's something that I've been intimately involved in. I just wanted to let everyone know about this new effort. So ALS Focus, it's led by patients and caregivers, and it's a survey program. And it's focused on asking people with ALS and people who are current and past caregivers about their needs and then their specific burdens. And so the goal is really to get as much individualized information as we can throughout the disease journey, and then use this and the results from these surveys to benefit the whole ALS community. And so this is something that's being done in partnership with investigators at Massachusetts General as well. All of the data that's collected is de-identified, and importantly, the results of the surveys are shared free of charge. So if you want to look this up, you can just Google ALS Focus, and that should pop right up. And you can sign up and participate in the surveys as you wish. And I would just highlight that the next survey will be released sometime this month. And they are designed so that they don't create an extensive amount of time. So the goal is 10 to 20 minutes for these surveys. And there's an initial demographic survey that you can fill out, and you can do that at any time. And then as surveys come around, you can decide to participate if you are able. And so I'll end with just saying, what does the national research portfolio look like this year and beyond? So some of the questions that we're asking: how can research help people with ALS live longer? So we're funding preclinical testing for the lead drug candidates and trying to get those into that pipeline, and focusing on personalized medicine. How can research help people with ALS live better? So assistive technology is a big part of this, and telemedicine and telehealth are important components. And then how can research help us determine why people get ALS and how we can keep others from getting it? Validation of environmental risk factors, and so this is something that we've partnered with the CDC on, and then setting up genetic screening and counseling, really to try to continue to move these efforts forward. So with that, I would just like to thank you so much. I look forward to our question and answer session. And I've just listed here my email address. So if there's something we don't get to in the question and answer session, or if you just want to touch base on something, please feel free to reach out to me. I will do my best to get back to you right away. So thank you so much, and we'll see you soon.